Yoo-hoo! Hello everyone! Welcome to Teacher Aisyah class! Okay, how are you? Are you doing well in your biology subject? Why? Did not understand Meiosis well? Okay, let's learn topic 6 with me, subtopic 6.3 and 6.4, which is about Meiosis. Okay, in this lesson, we will explore the detail of what happens during the phases of meiosis. Okay, meiosis, sometimes called as reduction division. It is a type of cell division that produces gametes. Okay, meiosis is the process of cell division that occurs in reproductive organs to produce gametes that contain half the number of chromosomes, half one, of the parent cells diploid. By gametes, we mean sex reproduction cells such as sperm cells in male, which occurs in testes, and egg cells in females that occurs in ovary. The need for meiosis. Okay, meiosis form gametes through the process of gametogenesis and ensure that the diploid chromosome number of organisms that carry out sex reproduction is always maintained from one generation to the next generation. Meiosis also produces genetic variation in the same species and broken down into two stages of cell division called meiosis 1 and meiosis 2. Okay, meiosis 1 have 4 phases: prophase 1, metaphase 1, anaphase 1, and telophase 1. And meiosis 2 also has 4 phases: prophase 2, metaphase 2, anaphase 2, and telophase 2. Just remember the PMAT formula, okay? Okay, you need to practice drawing too, because it will help you to understand deeply and clearly. Okay, while we draw together, I will explain to you in this video, and you need to listen to me and draw the chromosome clearly, okay? At the beginning of cell division, DNA condenses tightly into an X-shaped structure known as chromosome. Each side of an X-shaped chromosome is genetically identical sister chromatid, forming a sideways V-shape. In the middle of a structure, called the centromere, joins the sister chromatids together. If you want to draw the chromosome, please don't forget the centromere. Okay, different types of organisms have different numbers of chromosomes. Every cell in our body is called as somatic cell, except our gametes. Okay, human somatic cell has 23 pairs of chromosomes for a total of 46. It's just like how we count the pair of shoes. We have one pair of shoes, but we have actually 2 shoes, right? One right and one left, isn't it? Understand? Okay, let's look at what happened during meiosis 1. Okay, prophase 1 starts with the diploid cell. Its chromatin contains 2 uncoiled spread out sets of chromosomes, one from each parent. After the DNA in the chromatin replicates, it condenses into the more familiar X-shaped chromosome. Okay, remember the X-shaped chromosome. The replicates DNA is the same in the identical sister chromatids of each chromosome. In a process called synapsis, each chromosome pairs up with and binds to its corresponding homologous chromosome, forming a tetrad. Okay, what is tetrad? Tetrad is the group of 4 sister chromatids in a paired homologous chromosome. Remember that. What is tetrad? Tetrad is 4 sister chromatids. Okay, chromosomes contain genetic information called genes. These genes were inherited from each parent, and different versions of the same gene on each chromosome are called alleles. In a process called crossing over, chromatids from each homologous chromosome exchange segments of alleles, also called recombination. Crossing over randomly happens on every chromosome, resulting in different gene combination. This explains why every gamete is genetically different from every other gamete. Crossing over results in genetic variety of offspring. This is why children are different from their biological parents as well as from their biological siblings, just like us. Continuing on with Prophase 1. The nuclear membrane disappears, the centriole moves to the opposite ends of the cell, and the spindle fibers fan out from them. See it clearly, okay? Watch this video. Okay, next in Metaphase 1, the homologous chromosomes line up at the equator and attach to the spindle fibers from opposite poles. But remember, please alert, the spindle fibers attach to the centromere, okay? The centromere, the spindle fiber attached to the centromere. During anaphase 1, spindle fibers separate the homologous chromosome in each tetrad and pull them to the opposite poles of the stem, okay? Pull them. The spindle fibers pull the chromosome. Okay, here teacher wanna highlight to you the animation how the chromosome have been pulled, okay? Please take a look. See, do you notice how the chromosome move? The animation, the shape, the way it pull like this. Not the opposite, okay? It pull like this. This is spindle fibers and it pull the chromosome away, okay? It's just like when you pull a string where it attached in the middle of the card or the paper. Can you imagine? It will move like this, not like this. Okay, next, the cell enters telophase 1 with one chromosome for each homologous pair at separate poles. However, each chromosome still consists of sister chromatids. Keep in mind that each chromosome's sister chromatids are no longer identical because of the allele exchange that happened during the crossing over. Then, spindle fibers disappear and the nuclear membrane reform around the chromosome. Wait, we stopped here for a while. What is this called? It is cytokinesis. What is cytokinesis? Cytokinesis is the physical process of cell division, which divides the cytoplasm of a parental cell into two daughter cells. Excellent. Okay, let's proceed. Finally, cytokinesis occurs. Meiosis 1 ends with two genetically different haploid daughter cells. Each haploid cell contains only one set of chromosomes consisting of paired sister chromatids. Both cells now at the next stage, meiosis 2. However, unlike meiosis 1, DNA does not replicate before meiosis 2 begins. Once again, in prophase 2, the nuclear membrane disappears and spindle fibers fan out from the two sets of paired centrioles. During metaphase 2, the chromosome in each cell line up at the equator and attach to spindle fibers from both poles. Okay, during anaphase 2, the sister chromatids of each chromosome separate and move to opposite poles. Once the sister chromatids separate, they are called chromosomes. Finally, during telophase 2, the spindle fiber disappears and the nuclear membrane reforms, and cytokinesis occurs in both cells. Meiosis 2 ends with the four genetically different haploid cells, each containing only one set of chromosomes. Now, let's see some key points to remember about meiosis. First, it begins with a diploid cell. Meiosis only produces gametes, and gametes are genetically different haploid cells. Sperm cells in male and egg cells in female. Next, meiosis has two stages of cell division called meiosis 1 and meiosis 2. During meiosis 1, what happened? Homologous chromosomes separate to produce two haploid cells, each containing chromosome in the form of paired sister chromatids. In meiosis 2, the sister chromatids separate in both cells, becoming individual chromosomes. Cytokinesis of these cells produce four genetically different haploid gametes. Okay, well done. But wait, we not end this topic here. Without further ado, let's continue our learning session with subtopic 6.4, issues of cell division in human health. Okay everyone, take a look at this picture. What we call it? We call it as phases of cell cycle. The cell cycle is controlled by a special control system at each G1, S, G2 and M phase to ensure proper division of the cells. However, uncontrolled cell division sometimes can lead to the formation of tumor. Tumor is divided into two types, which are benign tumor and malignant tumor. A benign tumor is not dangerous and can be removed surgically. A malignant tumor is also called as cancer. Cancer is caused by several factors such as radiation, chemical substances, carcinogens, genetic factors, and also bacteria and viruses. This will cause the cell to divide continuously and develop into a tumor. The cancer cell will spread and destroy the normal cell around it. This condition will affect the function of the tissues around them. Cancer that is not identified at the early stages can cause damage to the organs and finally death. For example, the breast cancer. As you can refer to your Form 4 Biology textbook, page 108, you can see the example given, the development of breast cancer. Furthermore, any abnormality during the division of meiosis can also cause genetic diseases. I will give one example of the genetic diseases, and it is a Down syndrome. Before I explain further, let's watch a short video about Down syndrome.
Today's topic is Down syndrome. Down syndrome is a genetic disorder and the most common autosomal chromosome abnormality in humans, where extra genetic material from chromosome 21 is transferred to a newly formed embryo. These extra genes and DNA cause changes in development of the embryo and fetus, resulting in physical and mental abnormalities. Each patient is unique, and there can be great variability in the severity of symptoms. Causes: human cells normally contain 23 pairs of chromosomes. One chromosome in each pair comes from your father, and the other from your mother. Down syndrome results when abnormal cell division involving chromosome 21 occurs. These cell division abnormalities result in extra partial or full chromosome 21. This extra genetic material is responsible for the characteristic features and developmental problems of Down syndrome. Any one of 3 genetic variations can cause Down syndrome. Trisomy 21. About 95% of the time, Down syndrome is caused by trisomy 21. The person has 3 copies of chromosome 21, instead of the usual 2 copies in all cells.
This happens because the spindle fibers fail to function during anaphase 1 or anaphase 2. As a result, the chromosome fails to separate, or we call as non-disjunction. Gametes would have an abnormal number of chromosomes, 22 or 24 chromosomes. If fertilization between a normal gametes, 23 chromosomes, and an abnormal chromosome, 24 chromosomes occurs, the zygote will carry 47 chromosomes, which is an abnormal condition because it should be 46, not 47, right? This syndrome can cause mental retardation, slanted eyes, and a slightly protruding tongue. A child with Down syndrome displays certain characteristics such as stunted body growth and mental retardation. Wow, there's so much information and new knowledge for you. Hopefully you enjoyed the learning session and understand the topics. Have a nice day, good luck, and thank you!